Good afternoon, students. It seems like the snow and the weather is getting the best of our class. You know, I'm deeply saddened because I love teaching this subject. Well, what I want to do is add some extra notes to your chapter 3. This stuff has been posted for since last week, probably the week before last. And I wanted to give you some last minute notes before your test next week. I'm not changing the test date. It will still stay the same. But hopefully we will go to class tomorrow, which will be Wednesday, and Thursday in lab, and I can review over anything that you may have questions with and need clarity. I'll also do another review session the following Tuesday to make sure that everyone is feeling quite comfortable about the test. So let's go ahead and dig right in with cellular level of organization. So we know the cell is the functional unit of life. There are about 3 trillion cells in a human body. The main point I want to pull out is eukaryotic cells versus prokaryotic cells. Eukaryotic cells are membrane-bound organelles. They have membrane-bound organelles. That's where you see things like the mitochondria, the endoplasmic reticulum. You'll see those organelles. Whereas in prokaryotic cells, you're not going to see that. What you will see is some DNA, and that's about it, and a semi-nucleus. So those are the things that I wanted to really point out, the difference between the two. Now, the cell membrane, which is very, very important. Sometimes you'll see it called the plasma membrane. The reason it's so important because it protects the cell. It pretty much acts as a bouncer. So think of the cell as a club and the plasma membrane, cell membrane, as the bouncer to decide who goes in and who goes out. And because of this deciding of who goes in and who goes out, we know that the membrane is semi-permeable. Depending on what you are or who you are determines whether you can get in or get out of the actual club cell. So one of the things that I wanted to show in this diagram were the phospholipids. So here we have this entire structure is made up of phospholipids. So we see at the very top, this yellow circle right here is the head of the phospholipid, and that is called a phosphate head. So this is a phosphate head. Now the squiggly lines here are actually called fatty acid tails. Now if you were to look even closer up under the phosphate head, you would find a glycerol backbone, and it does exactly what it says. What the glycerol backbone does is serve as a point of attachment for the tails in the actual phosphate head. Now some more properties that are very important that you understand is hydrophilic and hydrophobic. Hydrophilic means water loving. So your phosphate heads right here love water, because what we will later find out is the cell is comprised of completely, the majority of the cell is composed of water. So these heads actually love being in the water. So they are hydrophilic. Now, inside we have the hydrophobic region. Phobic meaning of fear or afraid. So they're afraid of water. So they want to compact as closely as they can to stay away from as much water as possible. What happens if they come in contact with water? They're not going to shrivel up and just lose entire function, but they prefer to be in as closely as an impact area as possible to stay away from water. So this diagram pretty much keeps them pretty shaped. So what happens if we actually put them in water? The tails, they shrivel up. They won't lose function, but they will shrivel up, and the heads get closer and closer together, just as a sort of protection. Okay, so now let's look at, here's the famous word, amphipathic. Amphipathic, very, very important word here. And what the amphipathic means. Amphipathic right here, or what that means is that the area has 2 places where they both have hydrophobic and hydrophilic parts. Remember, the hydrophobic will be those fatty acid tails. As you can see how closely they are linked right here, and you see the heads are at the very, very top because they absolutely love being in the water. Okay, and the cytoplasm, which we'll get into, is, you can see, full of water on both sides, so the tails are trying to hide from that area. Now, what I do want to talk about is, again, talking about the bouncer of the cell. Semi-permeable. What are the characteristics you need to actually get inside of this club? What you have to be is a small, non-polar molecule. So examples of small non-polar molecules would be like oxygen, which would be O2, and carbon dioxide. They can swim through this area very, very easily without any help. So it's very easy for them to pass through this membrane. The bouncer has no problems. Okay. Now, some things that are small and polar, like water, they can pass through, but it's at a much slower rate. So the ideal thing to pass through with little help, well, no help at all, would be small non-polar molecules. But small polar molecules like water and ethanol can go through. Anything big that's trying to go through the bouncer will reject it, and it's going to have to find another way to actually get in. Now, here we go. We see cholesterol is actually placed throughout the cell, and it's very, very important because it keeps the model. You'll see the name fluid mosaic model. It keeps the fluidity going, so it keeps it from solidifying at room temperature. And what that actually means is the membrane's ability to move through the watery or the aqueous conditions of the cell is decided by three things. And those three things are temperature, cholesterol, and unsaturated fats. So the key thing is low temperature and fluidity. If there's low temperature, then the fluidity is going to be very low as well, because these little heads and tails are going to huddle together because there's not enough energy, so they're not going to move much. And if you think about it, when you're extremely cold, you usually try to sit down somewhere and get where something is warm. You want to cuddle up because it's so hot. I'm sorry, because it's so cold. Now, the opposite happens with high temperature. If you have high temperature, fluidity increases a great deal. You think about when it's hot, you don't want to be closely bound to anyone. You want to get away as much as possible. Cholesterol, again, since we see it here in the structure, if you increase the distance, the fluidity increases. So the more cholesterol is spaced out, we have more fluidity. Because they actually want to be attached. Now, saturated fats, and we see all types of commercials with saturated fats, so which are what we eat. Now, saturated fats are usually associated with heart disease, atherosclerosis, the hardening of the arteries and the blood vessels. These are where you also see your LDLs. For those of you who have gone and gotten your cholesterol check, we know that's the lousy, lazy cholesterol. We see it more so in processed meats. It's very, very easy for these saturated fats to kind of stick together. And when they stick together, the fluidity decreases. And that makes sense because if you think about any diseases that deal with the heart or a consumption of large fatty acids, it's very, very hard for everything pretty much to move, whether it's blood going through the vessels or if it's nutrients trying to get through the vessels as well. It's very, very hard to do that. Unsaturated fats, which we know are the very good fats, we see those in olive oils, we see those in your really, really good oils, your palm oils, and they they create a high level of fluidity. You'll also see it again in avocado and fish oils, those lovely cod liver oil pills that our parents made us take that were awful. But they have an importance in keeping the fluidity of the cell at a high rate, so everything is moving in accordance to where it should be. Okay. Now, let's move to the next part. Let's actually talk about the organelles of the cell. We'll actually talk about that. First thing with the organelles, well, I want to start with the cytoplasm. Cytoplasm is very, very important. It is present in both eukaryotes as well as prokaryotes. 80% of the cytoplasm is composed of water, and most of your cellular activities occur here. For instance, glycolysis and cell division, and that will be very important once we start talking about transcription, translation, and replication. Now the cytosol, which you see, so we have the cytoplasm here, and then you'll see the cytosol, which I'll show a much better picture in the next slide. And what the cytosol does is it does not contain any membrane-bound organelles. So the cytoplasm is going to have all of those organelles we need plus the cytosol. But the cytosol by itself does not have any organelles in them. Alright, so let's get to ribosomes. Now, ribosomes are very important. We often see or often always hear that ribosomes are the site of protein synthesis, which is true. Ribosomes can be found free or bound, meaning they can be floating around in a cytoplasm or they can be bound to another organelle. And that other organelle, of course, would be the rough endoplasmic reticulum. So we'll actually get a chance to see that. Now, the rough endoplasmic reticulum, we say again, it synthesizes protein. But to be more specific, what it actually does is it's a manufacturing site. If you all think about the old cotton mill in Henderson, it's an old manufacturing site. It puts everything together. It's going to manufacture proteins and help export them to other organelles. So it's going to take it to a place where it knows that it needs to go through a better process. Let's see. Once we drop down to the smooth endoplasmic reticulum, which is the complete opposite of the rough endoplasmic reticulum. The rough endoplasmic reticulum is rough because ribosomes are attached on them. The smooth endoplasmic reticulum is smooth because there is no bumps or any other substances on top of them. The smooth endoplasmic reticulum is very important because it detoxifies drugs or other harmful substances. It also detoxes lipids and steroids, especially ethanol, and we know ethanol as alcoholic beverages. And what you'll find interesting is scar tissue. Usually you'll see scar tissue in people that have cirrhosis of the liver, because the liver is a very, very smooth organ. If you've ever seen it up close, very, very smooth because it is covered in smooth endoplasmic reticulum. But when people continue to drink, what happens is it almost burns the smooth endoplasmic reticulum off and causes scar tissue, and the body is no longer able to detoxify at a degree that it was once able to do. So you have that rough scar tissue. So I encourage you to look up a picture of a liver that's going through cirrhosis and a normal liver, and you'll see a huge difference. You also can see that scar tissue from people who have gone through a severe overdose of barbiturates, and once you get into nursing school or your professional school, you'll actually get to learn all those lovely names of all of those barbiturates. Next part, let's go ahead and get into the Golgi complex. The Golgi complex, or you may see it as the Golgi apparatus, is the UPS of the body. And the reason I call it this is because it can modify proteins, it sorts them, and it ships them. The Golgi complex is very, very important in making sure that everything gets the way it's supposed to go. It has to show direction to where these proteins can function. The next part is lysosomes. Lysosomes, I call them the cleanup crew because they digest substances in the cell. Whether it's something worn out or a red blood cell that is past its date, whether it's bacteria or some type of virus, that's what lysosomes come in and do. And usually you will see your lysosomes in certain parts of the body, especially in sperm. If you see the head of the sperm, it is filled with something called acrosomes, and at the top of it, what happens is the lysosomes, which is full of acid, will eat away at the egg, and it eats away at that egg covering so it can go in and fertilize. Without the lysosomes at the head of the sperm, the acrosome, it would not be able to penetrate the sperm. So that is very, very important. Another important organelle are peroxisomes. They of course make peroxide. They break down large fatty acid molecules, very, very long ones. We talked about the word catabolism in the last chapter, how the catabolism is the breakdown of something very, very complex to something very simple. For example, if we were going to take a bowl of Cheerios, and how the body needs glucose, so it's going to break those Cheerios down into glucose, something simple that we can use. So it's going to take it from the carbohydrate and actually get glucose in them. Now, there is a disorder called that affects the myelin, and what the myelin is, it's in the central nervous system, and it helps to speed up the transmission of neurological messages. And when that is affected, what happens is it can lead to a vegetative state or even death if those signals or those messages are not getting to the correct places. It was actually a movie called Lorenzo's Oil that actually talked about that, and it was an X-linked disorder, and once we get into genetics, we'll actually talk about that some more. So what pretty much the cure was for that was to make sure that the child did not have any complex fatty acids. Everything that had to be very, very simple and already broken down. All right, the next one is the mitochondria. Mitochondria is the powerhouse of the cell. This is where all the energy is coming from. Very, very important. Another interesting fact about the mitochondria is that it does contain DNA. Usually we think of just the nucleus containing the DNA, but mitochondria also has DNA as well. Proteasomes break down faulty proteins, because we know it is the body's main idea is always to get everything in homeostasis. So there will be some times where things will break down, and proteasomes, pretty much like lysosomes, will make sure that they break down and get rid of anything that is not wanted. All right, the nucleus, as we can see here, this is the big lovely nucleus. It contains most of the cell's DNA. And the reason that it says most of the cell is because, remember before I just said that the mitochondria also contains DNA. And what we can see in here, we'll also see some chromosomes. You see those large axes. They are what's found inside of the nucleus, and we also know that it includes DNA. Again, the cytosol contains water dissolved solutes. Remember, you have the cytoplasm, which is a combination of the cytosol as well as the organelles. The cytosol by itself is just water, solutes, and chemical reactions. Another thing I wanted to add were centrosomes. Centrosomes are very, very important because they organize microtubules. And the purpose of microtubules, they're very, very, very important. They are the keystone in stability in the cell. They help to make up the cytoskeleton, which is pretty much the skeleton of the cycle that provides protection for everything involved. Now, the cilia are short hair-like structures used for the movement of fluids, but they're also found in the trachea, and is used to sweep mucus out. Oftentimes when you have some phlegm in your throat, usually what has happened is the mucus inside of your throat, along with the hairs, the cilia and on your trachea, will cough those pieces up. Now, cilia can be burned off due to aggressive smoking. Heavy smokers can actually burn those cilia hairs off, as well as those of you all who like to drink excessively hot items such as extremely hot coffee or tea. That will also burn the hairs off. We also see cilia again in the fallopian tubes, because the important part there is they're going to move the ovum from the ovary to the uterus. So very, very important again there. Now, if there is a defect here, what you will find is, you may have heard of women going through something called tubal pregnancy. Or someone will say, you know, she was pregnant in her tube, or she had a tubal pregnancy, or ectopic pregnancy. And what that means is there may have been a misfunction in the cilia. So remember, everything always happens at the smallest areas. So what has happened is the cilia is not able to move the ovum to the correct places from the ovary to the uterus so they can be implanted. Flagella is very similar to cilia, except it's very long hair, and its main function, of course, is movement. And we see flagella on the tail of the sperm. Now the interesting thing is, if for example something happens with the microtubules, you're starting to see a pattern how it will affect cilia. It'll affect the fallopian tubes, it affects the trachea. Well, a flagella, it would actually affect the sperm. So if the flagella if something has happened and there's a malfunction, what will happen in sperm is you will actually have no movement in sperm. So there's a defect. You will actually have male infertility because the sperm is not able to move. It is at a standstill, so it's not going to move. All right, the next one, diffusion, osmosis, facilitated diffusion, and active transport. All right, diffusion. Diffusion is very important. We'll see more and more of this over and over. Pretty much what diffusion is we're going to take areas. We're going to take a high concentration, a high solute concentration to a low solute concentration. So it wants to move from an area of high concentration to an area of low concentration. And I'll actually draw these out in lab class so you can see a better picture of them and how they actually work. Simple diffusion is very pretty much what it says. It is easy. It helps molecules will move throughout the membrane without any help. Example, hydrogen, steroids. We talked about carbon dioxide earlier. So they can move quickly through the membrane. I will call them the A+ group because it is easy for them to get into the bouncer. It is easy for them to get into the plasma membrane. The facilitated diffusion, again, does not require any energy, but it needs help. So it's like you have that one friend that has the hookup to help you get into the club. This is what facilitated diffusion is. It knows it wants to get in, but it knows it can't get in without the proper hookup. Someone has to help them get in. So that help comes through a form of a protein carrier or a channel. Okay, so they will pass through like glucose. Glucose cannot get in. It is not a A-list celebrity. So they have to have someone that knows someone to help them slide through the door. Then you run into active transport. And what active transport uses is they decide that they are going to pretty much bum rush the bouncer. They're going to go against the concentration gradient. Everyone has gone in pretty smoothly or with the help. They're going to run through full speed using all of their energy. Another way of saying it is they want to paddle uphill the river. They want to paddle uphill. They have to have some energy to do this because, of course, if you're going to exert all this energy to, or you're trying to actually get into this place, it's going to require a lot of energy. And one of those examples would be the sodium potassium pump and how that actually gets in. Now, before let's talk about hypertonic, isotonic, and hypertonic solutions. What a hypotonic solution, isotonic and hypertonic, this actually moves us more into osmosis because osmosis has to deal with the movement of water. A hypotonic solution, one way to see it is water is going to move into the cell, and because water moves into the cell will swell and eventually burst. What happens is, one of the things you must remember with water is water follows salt. So whenever you eat a bag of Lay's potato chips, instantly you're thirsty after eating them because the body now wants more, wants water because it's thirsty. Water always follows salt. Always, 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 always. So what happens is usually if we're looking at a diagram, let's say that water, we have a solution, and outside of the solution, let's say the solution right here. So let's say outside of here we have 10% salt. And let's say inside here we have 20% salt. Well, the water is going to go in wherever there is more. So if there's 10% salt here and 20% salt inside of here, the water is going to shoot right inside of here, because remember, water follows salt. Now with the isotonic solution, it means both are equal. There is no extra. You have the same amount salt here, so let's say it's 10% salt here and 10% salt here. It is equal. There will be no change in how it moves. The net flow will not change. Now with hypertonic solution, what happens is water is going to move out of the cell. And water moves out of the cell is going to do something to the cell. It's going to make the red blood cells shrink, and it's going to make it die. Another word for that is crenation. 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 And how that occurs is, let's say again that we have outside, we have 20% out here, 10% in here. So it's going to leave out. That water is going to move out of the cell, and it's going to cause this to shrink. So it is going to shrivel up. Okay. Next part, phagocytosis, pinocytosis, endocytosis, and exocytosis. So we have three different types. We have, let's start with cell. Actually, I'm going to go back one slide. We're actually going to start with receptor-mediated endocytosis. The whole purpose of endocytosis is when a cell is going to engulf some extracellular material. So something is dissolved or something is eaten. So that's what it wants to do. So with receptor-mediated endocytosis, it's going to take up cell ligands. And all ligands are molecules that bind to specific receptors. For example, LDL, we just talked about earlier when we were talking about membrane fluidity. It's a saturated fat. It is lazy. You also have iron and you have vitamins. All of those are specific receptors. So let's use iron. So iron is transported by protein transferrin. Very, very important. Now, there are cells. These cells have receptors for transferrin on their surface. So it's almost like a marker saying, hey, here we are. Now, when the receptors see the transferrin, it's almost like they're flashing a light. Like, hey, here we go. When the receptors see that, they grab it. And once they grab it, they engulf it by endocytosis. Once they finish it, then they release it, and the iron is released in a cytosol. So all it does is pretty much a cell cycling. It's a cell signaling process. Okay, let's move to phagocytosis, which is cell eating. So it eats solid particles, especially bacteria. Bacteria is we'll use bacteria for this example. So it's going to take bacteria from the ECF, which is the extracellular fluid, and the particle is going to bind to the plasma membrane. So again, we're going to look for a receptor. A receptor is going to be on a phagocyte. And once the receptor on the phagocyte binds to the particle, it kind of snatches it up, and you'll see a better diagram in your book. The particle is going to be surrounded by a pseudopod. And then once that phagosome fuses together, they fuse the phagosome with a lysosome, and they destroy it. So just to repeat that again, you have the ingestion of a particular matter, we'll say bacteria, from the extracellular fluid. and that particle is going to bind to the plasma membrane. Once it binds to the plasma membrane, a receptor on a phagocyte is going to hold on to it. Once it holds on to what that bacteria is, pseudopods begin to project from around the particle and enclose around that membrane, and now it becomes a phagosome. Now, when the phagosome and the lysosome fuse together, you have phagocytosis, because that means it's going to destroy, it's going to eat that bacteria. And you see that more so in neutrophils and macrophages. So neutrophils and macrophages are pretty much where you see that. Now, a great exception where nature has decided to outsmart what the body does would be like salmonella. Because what salmonella does is it creates a protein that keeps the fusion away. So what that means is it's going to create this protein that refuses. It's going to be like a blocker. It refuses to let a phagosome and a lysosome come together, because we know if the phagosome and a lysosome come together, there's going to be a fusion, and what's going to happen is it's going to be utterly destroyed. But salmonella has figured out a way to pretty much remove itself from them. Next is pinocytosis. Pinocytosis is cell shrinking. Very, very interesting. There are no receptors present here. And what it does is just take droplets of the extracellular fluid. They pretty much take it up, or they drink it up. All right. The next part with exocytosis. So it does the complete opposite. Endocytosis brings everything in. It's going to bring everything through a vesicle, whereas exocytosis is going to release the material from the cell. It's going to push it out. So I encourage you to click on this YouTube video and watch that area. All right, the next part is be able to explain protein synthesis and be able to distinguish between transcription and translation. DNA, very, very important. DNA is deoxyribonucleic acid, and this is where the majority of our genetic information is here. It's composed of nucleic acids. It has a sugar, a phosphate group, and a nitrogen base. Remember, we saw the phosphate group before when we were looking at the plasma membrane. We saw the phosphate head and the fatty acid tail. So here we're seeing the word phosphate again. Now, what's interesting is the human genome has about 3 billion base pairs to make a human. So it takes a lot to make you all you. Now, here are some of the bases. We have adenine, guanine, cytosine, uracil, and thymine. Okay, now what you will see is how they actually match up. We're going to put them into separate categories. So purines, pure as gold is how you remember what the purines are. Pyrimidines, cut the pie. C for cysteine, U for uracil, T for thymine, and PY pi for pyrimidine. If you remember one, you remember what goes in the other groups. Most people tend to remember purines, pure as gold, knowing that A is adenine and G is guanine. So whichever way that you choose to remember them. Now, these are how these are paired up. This is how this is paired up. In DNA, nitrogen bases are always paired A to T and G to C. Always, 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 always. Now, when you switch to RNA, the T is removed and you have U that replaces T. So it goes A to U and G to C. And we'll go through some examples of how to put this together. So if we look right here at DNA, we see what happens with DNA. In order to actually get it to replicate, we know it has a helix. It has a double helix. Let me see if I have a picture. Did I have a picture? Okay, I don't have a picture of this. But DNA, you know, is like a double helix. It looks like a winding staircase. So we have to separate those two. So when we separate those two, we end up with 2 strands of DNA. So this is why we have a strand here and a strand here. So what we see is G goes to C, C goes to G, A goes to T, A goes to T, T goes to A, C to G, T to A, and A to T, and so on and so forth. It keeps going. Keeps going. Now, that changes when we get to RNA. RNA, we will not have that T anymore. So you'll actually start to see a U. Okay, anytime you see a U involved, you know it's RNA. Okay. Now, the importance of protein synthesis. This is the process in which the cell builds proteins. And it's a pretty intensive process, but for all purposes, I'll make it pretty simple. What happens is you start off with DNA replication. And what DNA replication is, we start off with DNA replication, and what it is we're going to pull apart two strands of DNA. We're going to pull those apart, and once they're pulled apart, let's say we want to copy some more strands to make some more cheek cells or to make some more eyelashes. So we can just do that with DNA. So we just pull them apart, and the way we pull them apart is using things like heat, things like salt, aggravating it, things like that will actually pull it apart. So once we get that part done, then we have to actually get to the protein area. So if we want to make keratin, and keratin is a protein in your hair. So what we have to do is go through a process called transcription. And it's very, very important to know that transcription is going from DNA to RNA. And you must know that it occurs in the nucleus. This is very, very important. So remember earlier we were talking about the two strands of DNA from replication. One of those strands is going to be used to make a template. Okay? And when we use that template, we're going to use 3 other parts of RNA. We're going to use messenger RNA, we're going to use ribosomal RNA, and transfer RNA. And again, messenger RNA is the synthesis of a protein, ribosomal RNA makes ribosomes, and transfer RNA is going to bind it to the amino acid. Okay, so what happens here is the RNA is going to pick up where DNA left off, and it's going to transfer. It will actually transfer some of the information from the DNA into the RNA. And when we get in class, I'll actually be able to give you some examples. I actually have an assignment for you on that. So this is long, whole lot of words just to say what I was saying earlier. What we do is we're going to, for transcription, remember transcription is going from DNA to RNA. We're going to use some type of enzyme, or we're going to use heat or temperature to unzip the DNA. So we're going to break the DNA apart so we can see those base pairs, those A, G's, T's, and C's. Okay, once we do that, then we're going to take RNA and we're going to correspond them via RNA. We're going to do, for example, if I had A, G, T, C, then for my RNA strand, instead of my A going to a T, my A will go to a U. So if it's ATGC, it's going to be the A goes to T, A goes to U, G goes to C, C goes to G, T will go to A. And again, we'll go over plenty of examples in lab class. Okay. Now, once we get through transcription and we have the outline of transcription, we'll go through something called translation. So we're actually going to leave the nucleus. We're going to travel to the cytoplasm. Again, another video shows a really good example of how it works. So we're going to go in this process, we're going to go from mRNA to the actual protein. Okay. Explain cell division, mitosis, and meiosis. So mitosis, the best way to say it is asexual reproduction. It is also it's asexual reproduction. It also deals with anything other than a germ cell. And what a germ cell is a sex cell. So anything other than that. So it could be nails, it could be hair, anything of that set. Now the important piece with mitosis is you start off with 2 here, and because there's no mixing of sexual chromosomes, there's no mixture of genetic information, you end up with 2 identical sets in 2 nuclei. So we start here, we have 1 here, and as they go through the process, you have 2 identical sets. And those 2 identical sets are actually called diploid sets, okay? Because there's 2 sets. They are called diploid sets. All right, let's move to the next part. So there are 3 steps in mitosis. Well, let's say 3 steps in interphase. Let's start there. So interphase is 90% of the cell cycle occurs here. What you have here is G1, you have the S phase, and G2. So G1 is growth. We're starting off pretty small, and the cell is going to expound. S is where DNA is synthesized, and by G2 the cell is pretty huge. Now those steps aren't considered part of mitosis. Some people group them together. They really don't. Most people won't consider mitosis until you get into the actual PMAT stage. Okay. So let's move to the next slide. Prophase. Prophase, chromosomes condense and the nuclear envelope starts to disappear. This is very, very important. It's one of the factors in actually pointing out what prophase looks like. You're going to see the nuclear envelope start to disappear. You'll see breaks around the little area. Metaphase is pretty easy. Chromosomes will meet at the middle. You'll see them come to the, if you think about the equator on the globe, you'll see them come to the equatorial line. Anaphase is when your chromatids will start to pull away. I'll call them anaphase anti because they're going to go against each other, so they're going to be separated. The next one is telophase. Telophase is the opposite of prophase. So if you know prophase, you're going to know telophase because you're going to see the nuclear envelope start to assemble again. And what you see in telophase, you also see a piece of cytokinesis. And what cytokinesis is, the cytoplasm is going to divide into 2 cells. You'll see something, a pinching of the cell in the side. I'll go back to the previous slide. You'll see that pinching of the cell right there. That little indentation right there is called cleavage furrow. So make sure you know that little indentation is called cleavage furrow. So if we sit here and look at it, we see interphase here. See, when we get to step 2, we see prophase. You see that nuclear envelope has started to disappear. There's spaces in the line. It should be a complete line, but within prophase, we know that it starts to split away, and you will not be able to see it come the next step. You see the centromeres in the middle. And you see the centrosomes, which are these up here, are starting to appear. They started up together here, and as you see here, they're splitting apart. Those are called your centrosomes. And these are very important because they make your microtubules. And remember again, your microtubules are what make your cilia and your flagella. So this is the part of where it all begins. It begins in prophase. Now if you look here at metaphase, what you'll see is they have lined up in the middle. They've lined up in the middle. And you see that your centrosomes up here have moved all the way to the side, and you no longer see a nuclear envelope. The nuclear envelope is completely gone. When you get to step 4, the anaphase, you see the chromosomes have split apart and they're on opposite sides, so they're no longer joined anymore. And you can see the centromeres are starting to split out even more. By the time you get to telophase, you see that nuclear envelope is starting to reappear again, and your centrosomes have pulled back. You don't see the extensions as large anymore. And you see that little indentation right there, which will be your cleavage furrow. And then the cell cycle will start all over again. Now, with meiosis, meiosis is very, very similar to actual mitosis. Meiosis deals with germ cells, so it's going to deal with sex cells. So instead of having a diploid number, you're going to have a haploid number. There is going to be some exchanging of DNA information. Now, this pretty much ends lecture. I have plenty of diagrams that I'm going to draw. This program of recording is now going to allow me to draw them as well. So what I will do is go over this with you in lab. So tomorrow, be prepared to ask me any questions you have on chapters 1, 2, and 3, so that we can get through those things, because I will go over these things again and make sure you have a good grasp on them, and then we will start on chapter 4 as far as the lab part. If you have not had lab with me, then we will be working on lesson 3 in the book, which talks about I think it's lessons 3 and 4, where it talks about mitosis, as well as talking about what actually happens in hypertonic and isotonic solutions. So that will be exercises 4 and 5. Exercises 4 and 5 will be done if you did not have me for lab last week. Otherwise, I will see you all. If you have any questions, please send me an email. I do answer my emails, more so my personal one. Moodle, it takes me some time to get onto. I don't have that one readily accessible, but email me with any questions. All right, have a good evening and stay warm.